Hi, I'm Yasmine Messaoud-Nacer, postdoctoral fellow at the Institute of Molecular Genetics of Montpellier, a research unit of the CNRS and University of Montpellier in France. Today I will present a few glimpses of our study where we report a new role for the DNA-PK DNA damage response protein in the regulation of type 1 interferon response. But first, let me give you some context. It is well established that accumulation of double-stranded DNA in the cytoplasm activates the enzymatic activity of the cGAS DNA sensor. cGAS activation triggered the production of a second messenger called 2' through' cGAMP. This cyclic dinucleotide then docks onto the STING adapter protein. Triggering the formation of the STING signalism that orchestrates downstream activation of type 1 interferon and inflammation. Signaling axis is therefore essential in several contexts, such as response to pathogen infection. Despite its central role, no broad-acting direct inhibitor of 2' through' cGAMP that operates in a cytosol has been identified in mammals so far. We identified that DNA-PK directly interacts with 2' through' cGAMP through its kinase domain, alleviating STING activation and ensuring cyclic dinucleotide-associated signal attenuation. To demonstrate the interaction, recombinant DNA-PK was immunoprecipitated using a DNA-PK-specific antibody prior to incubation with 2' through' cGAMP. Then we carried out the release of bound 2' through' cGAMP, which we then quantified by ELISA. Next, we performed molecular modeling analysis to identify the putative binding region. Using the resolved crystal structure of human DNA-PK and 2' through' cGAMP, Doken experiment predicted that the 2' through' cGAMP docks directly into DNA-PK catalytic bucket. Afterwards, we showed that interaction of the 2' through' cGAMP with DNA-PK affect the biological activity of 2' through' cGAMP. To address this, control and DNA-PK knockout T98G cells were transfected with 2' through' cGAMP for 6 hours prior to gene analysis. An enhancement in inflammatory gene expression, interferon beta and CXCL10 can be appreciated in the absence of DNA-PK. Finally, we demonstrated that the negative role of DNA-PK on STING agonists translates into a biologically relevant functional response. As an example, human primary macrophages were treated with 3' through' cGAMP in combination with DNA-PK inhibitor, the NU7441, prior to infection with VSV. By combining STING activation and DNA-PK inhibition, we promote the establishment of a higher antiviral state, as shown by fluorescent microscopy and quantification of percentage of infected cells after 24 and 48 hours with both 0.5 and 5 MOI, supporting the role of DNA-PK in the negative regulation of STING agonists. This study provides critical insight into regulation of cyclic dinucleotides and highlights the implication for the development of STING-targeted therapies. We would like to thank the platform that we used on the campus, as well as the founding organisms, and thank you for your attention.